and Climate webinar, and everyone guys has been kind enough to present it, and it's going to serve a dual purpose for us, so we'll get to learn about the Florida Coastal Everglades long-term ecological research program and some potential connections with our project. So that's basically all I got to say, I think. And
thanks. Yeah, thanks everybody for coming. And it is totally a dual purpose presentation today. I know we have a lot of folks here from the WSC project, as well as the LTER folks who may be new to the program or want to be hearing updates on what we've been up to over the last year or so of the new, we're in our third iteration of this program, the third 6-year round. And so some new exciting things are going on that have relevance for the FCE, or for the WSC, and also for new folks who are thinking about or already come in newly to the LTER program. So I'm going to give you a little overview, a little bit of history, and then some updates on the newest, coolest stuff that's coming off the presses. I wanted to start with just an overview of who we are. What is this LTER program? How is it constituted within the LTER network? So who we are, we're based at FIU, of course, but we have 20 other institutions that we work with, collaborators from universities, as well as other institutions representing our other collaborative funding foundations and agencies, as well as different programs that we've been interfacing with to try to reach out to the communities, both locally and more broadly, building programs in order to really have a foothold and outreach into this community. We have 86 collaborators, 80 students. I think we're probably going to have a whole lot more pretty soon here, and 20 staff members. And we were added to this big LTER network in the year 2000. The network itself has been around since 1980. And I just wanted to tell you a few things about this network, because it's really important that the FCE LTER is a part of this in trying to understand the kinds of things we do here and the broader context for them. We do have 26 sites distributed around the United States, a couple in Antarctica and in the Polynesian island there. We have 2,000 scientists distributed around these LTER sites. And again, the network has been around for 3 decades now. The sites that have been around the longest have these incredible 30-year data sets that really inform us a lot about long-term ecosystem changes of the types that are really hard to capture over typical funding cycles that are normally 3 to 5 years, right? So the LTER is really at the heart of informing ecological theory relevant to the kinds of changes that we normally see in natural ecosystems that happen over long periods of time. Our sites span just about every kind of habitat. In fact, they're intentionally distributed in all of the major ecosystem types in North America and these other U.S. territories, from agricultural lands, tundra, barrier islands, coastal lagoons, hot and cold deserts. The estuary, wetland, and coral reef systems were brought on all together in the year 2000 when FCE began. Grasslands, kelp forests, lakes, open oceans, savannas, streams, and even urban landscapes. There's a newish kind of focus on urban ecology within the LTER network and making sure we understand the long-term dynamics in these cities. Our guiding principles throughout the network really are to, gain this knowledge of the diverse array of ecosystems over the spatial and temporal scales that we know matter. We also are really geared towards synthesis, probably more now than ever, taking the results from site-based, place-based research and linking them more broadly to what's going on in other places to inform ecological theory. The LTER network is really at the forefront of developments in information technology for the field of ecology. So we're really all about making sure our information gets out there, is broadly distributed and ready for others to use. We have, in addition to all these information resources, all kinds of other archives. So we've got the data archives, but also specimen archives, and samples that are around for future use. So that's another big goal of the network, is to create that legacy of information. We are also all about training, of course, at the graduate student level, but also dipping down into the K through 12 communities through big education outreach type programming. And our outreach, in addition to including the public interface with high schools and grade schools, we do have all these various partnerships with resource managers and policymakers and the broader ecological community to make sure that the science that we're discovering here is moving out to the communities that can use it for various both educational and scientific purposes. So because there are so many sites in the network, and we are all about making sure that what we learn about our place is linked to other places, we do have an adherence among all the sites to 5 core themes. And these started back in 1980 when the first sites came on, and we're all continuing to make sure that we address these 5 core areas, and those include patterns and controls of primary producers, so plant biology, spatial and temporal distribution of population selected to represent trophic structures, so moving on up through the animal world, patterns and controls of organic matter, accumulation in soils, patterns of inorganic inputs and movements of nutrients, so biogeochemical cycling, and disturbance ecology is another big one, making sure we understand how to use these long-term data sets most effectively to capture the effects of disturbances. Most of the LTR sites have adopted a network-wide initiative to really more deeply understand connections with human systems. So we have a common socio-ecological framework that our research is embedded in, and you'll see that stemming through most of what I talk about and relevance to our LTR site, which is of course embedded in a human landscape. We can't understand the Everglades without understanding the people in South Florida. Cross-site research and synthesis, again, a big goal of what we do. And it's important to know that we're not unique in the United States of having an LTR network, but in fact there's LTR networks throughout many countries all over the world. There's an international LTR network of sites that we're in frequent contact with and that have similar goals to the US network to even further build that ability to connect and make broader conclusions from the science that we're doing. So from here, I do want to focus mainly on what we're doing down here in the FCE. We do have these research, education, and outreach goals in common with the network. Our research is focused on long-term studies of freshwater availability and climate variability, how these interact with the biology, particularly in the coastal zone of the Everglades, and I'll get into that more deeply in a couple minutes. Our education, again, is focused on graduate student research, building the capacity for leadership and research among our graduate students. You guys are producing the bulk of the research findings that I'm going to be talking about today. We also have the Schoolyard program that reaches into the K through 12 communities, both to the teachers and to the students, bringing them into our labs. We're going out into classrooms and making sure that we have as broad a footprint in developing environmentally literate communities of folks moving up through grade school and into college eventually. Our outreach is not only through this educational mission, but also we have an FCE, a lot of decision makers, research managers, and agency scientists embedded right within the program. They help us figure out what science to do, how to do it, and how to make it publicly relevant. And so that's a really big hallmark of FCE. And again, we're making our information public as broadly as we can through various communication means that I will talk about too toward the end. I do want to focus on the research aspects of our program. That's what I'm going to spend most of the time talking about, but I'll have a few bullets at the end about what we do in terms of education and outreach more specifically. So our central theme is the changes in climate and resource management interact to influence fresh water supply, biological processes, and human dynamics. And we believe that this interaction of water supplies and decisions about the Everglades are played out most in a way that is most dynamic, perhaps, in this oligohaline ecotone, the place where sawgrass marshes integrate into the mangrove habitat in the southern Everglades estuaries. And so I want to move us back all the way to the beginning of the program, because I think to understand what we're doing today, it's really important to have this contextual sort of basis of from the past. And so before the FCE was funded, a proposal was put together based on what we know about what we knew at the time about how the Everglades ecosystem worked. And we were concerned about gradients of productivity in the system. And what we knew at the time was that the interior of the Everglades is incredibly nutrient depleted. And so productivity there in our sawgrass marshes is really, really low. But as you move toward the coast, you see these really incredibly productive fringing mangrove forests, okay? That you can see from these aerial imageries or remote images and from data that we had been collecting, particularly water quality data. You can see here the gradients in Florida Bay that have been shown in Joe Boyer's work for so long, that we have more phosphorus in the Gulf of Mexico than we have in the interior even of Florida Bay. And as you move from west to east, you see this declining concentration. Same thing, as we move up through the Everglades, we get less and less phosphorus. And so the hypothesis at the time was that ecosystem productivity in this landscape is really driven by the intersection of fresh and marine water supplies, the fresh water being a little bit more replete in nitrogen relative to phosphorus, and the marine end being the main supplier of phosphorus. So where we see the greatest productivity is where those two really primary plant nutrients come together. And so we set up this structure to study that. We established two transects, the Shark River Slough and Taylor River Slough transect, that run through the two major drainages of Everglades National Park. We established a working group structure, so all of our science is done within core working groups, and I'll go through what those are and why they matter to coming up with a more synthetic understanding of the Everglades. And just to mention the PIs at the time, the original proposal was written by Dan Childers and Joe Boyer, Jim Corcoran, Rudolph Jaffe, and Joel Trexler were really spearheading this effort to get the program off the ground. And here's an illustration of their hypothesis. So here's the freshwater to marine gradient represented here on the x-axis, and ecosystem productivity as a function of that gradient along Taylor Slough in red and Shark River Slough in yellow. And our hypothesis was that these freshwater inputs, low in phosphorus, high in nitrogen, perhaps relatively high in organic materials produced from within the system and moving down through, intersect with these marine supplies a little bit more replete in phosphorus. And we see in the Shark River Slough, where we had hypothesized that we would see a peak in productivity at that intersection. And in Taylor Slough, the absence of a productivity peak, because we have this expansive Florida Bay full of seagrass communities that are, and marl soils, calcium carbonate rich soils, that are really good at sequestering phosphorus. So that's why we expected not to see a peak in the Taylor Slough. Well, what we ended up finding, and I'll walk you through a little bit of the reasoning here in a couple of subsequent slides, but what we found was that we actually see a peak in the Shark River Slough ecotone more toward the marine end member, and we think that that's due to, or thought at the time that was due to the movement of phosphorus into this community driven by tides and storms. And we were surprised to see in the Taylor Slough ecotone this dry season peak in phosphorus availability and in the productivity of plants. So we were really curious about that. All of our data was showing this incredible peak in that oligohaline ecotone. And through advancing some hydrologic research through Renee Price's group, we were able to see that, again, affecting the Shark River Slough ecotone, we see a much greater tidal amplitude and an impact of storms coming from the west to the east and driving that phosphorus into that and that marine end of that ecosystem. But in the Taylor River Slough and also a little bit in Shark during the dry season, we see marine groundwater moving through this really kind of Swiss cheese bedrock, limestone bedrock. So moving very easily through that bedrock during the dry season because there's no fresh water coming through to counteract it. And it bubbles up, bringing with it phosphorus that is mobilized from the calcium carbonate. So Renee's work has shown that this salt water moving through the bedrock is really mobilizing phosphorus, especially in that dry season. And when we look at our productivity data, this is just annual net primary productivity of these major different groups, algae, sawgrass, mangroves, and seagrass, along the two transects from upstream to downstream, left to right there. And we see the peak of productivity in those zones that I mentioned, the coastal areas of SRS and Saltwater, and the ecotone of the TSPH transect. So that's a really fast summary of what we found in the first 6 years of the program. Moving into the next 6 years, we were envisioning a lot of restorative activity due to the Comprehensive Everglades Restoration Plan, the removal and of levees and the enabling of freshwater to move back through the system in a way more commensurate with what we envisioned would have been the case in the historic setting. So in the middle here, we have the current flows being all interrupted by our canal systems, and then the removal of a lot of those barriers to flow through the SERP plan. We anticipated that this would really have a big impact on the shape of these productivity curves in these two transects, right? If we have, so at the top here, I'm showing what we discovered in our FCE I program and FCE II, we hypothesized that more fresh water would be coming in at least to the Shark River Slough ecotone, because that was where we anticipated major bridging of the Tamiami Trail, the main impediment to water flow from the water conservation areas down into Everglades National Park. And that if that fresh water was, again, moving through the way that it historically did, we anticipated that the ecotone in Shark River Slough would move even further to the west, and in Taylor Slough, recovery of some water movement through some of the boundary projects that were commensurately being planned. Perhaps we would see an absence of that dry season productivity peak. So our hypotheses were all about that increasing inputs of fresh water would enhance oligotrophy, moving more nutrient depleted fresh water through the system is going to push that productivity zone out toward the sea, right? Increasing oligotrophy. At the same time, perhaps we're moving more organic matter through. And that organic matter, if it's the base of a detrital fluid web in the ecotone, which we had a lot of evidence for, then there may be stimulation of the food web by that organic matter flow. And finally, the third hypothesis was really related to hydrologic change in this landscape, that water residence time, groundwater inputs, and tidal energy are really interacting to modify the patterns that we see in the ecotone. And at the same time, we added a new theme of really focusing on human dimensions and understanding decisions that affect the movement of water and really the decisions around restoration planning that influence our ability to project what's going to happen within the heart of Everglades. I just wanted to mention that this is kind of how we are put together. If you're considering joining the program, you may want to look at these working group structures. They're a little different now. I'll show you the current configuration. But, you know, our research is done within these pillars that are similar to the core areas that I described earlier, the biogeochemical, primary production, trophic dynamics, and organic matter. And then we have these cross-cutting thematic areas that really are informed by the pillars and help us synthesize information. And those include this human dimensions work, hydrology, climate and disturbance, and modeling and synthesis. And so as we moved into FCE II, we anticipated the grand experiment that would mobilize more water into Sharp River Slough. And at the time, on the table was between 5 and 8 miles of bridging on Taniemy Trail. So we really thought that this was going to have an incredible impact on the Sharp Slough. And this is just an artistic rendition of what we thought the trail was going to look like. But this all evolved, and we ended up, as you know, with this 1 mile bridge very far to the east of the main flow path of Sharp River Slough. And I have an X over this 2.5 mile bridge further west, because that is what happened during that time frame of our FCE II program. But just recently, the governor of Florida has decided that perhaps we may have some funding to move forward with that. So that's hopeful news for the future. But we don't have this massive expanse that we had anticipated. And so while waiting around for something to happen in Sharp Slough, we needed to test these hypotheses in a little bit different way. And so we ended up looking at variability. Long-term data series are fantastic for that. We have wet seasons, dry seasons, wet years, dry years, to understand a little bit more about how the balance between fresh and rain water supplies influence productivity along these transects. We're all the time testing the antithesis of restoration, as restoration is delayed. And we can do some space for time substitutions because connectivity in Taylor Slough was actually being affected a little bit more than Sharp River Slough by some projects that were happening there. A little bit of, oh, shoot, this is not showing. Very strange. I apologize for this slide. It was meant to show background data on total phosphorus concentrations throughout the system, where we saw a huge effect of Hurricane Wilma moving through the system. So this is speaking to our ability to use events as signals of change in our system, and or as drivers of change in the system. And yeah, what we see, it's really too bad. I have no idea why these data aren't showing. But we see this huge peak, kind of mimicking the yellow peak here is the elevation of soils at a site near our marine end member of the SRS transect. And that huge peak in soils is actually coincident with Hurricane Wilma coming through, bringing up soils from Florida Bay and dumping them there in the mangrove forest. Those soils were full of phosphorus. They, over time, leached back out. Our phosphorus numbers are way up in the 20s and 30s parts per billion, which is really high for our estuaries. And they remain high because they're constantly being washed out as the tides come in and out. So, you know, one big storm event creating a long-term legacy effect in that ecotone. Okay, at the same time, we have long-term data on the productivity of our forests, driven by data from our Eddy covariance tower down at SRS 6, in this really tall fringing mangrove forest. We have in the blue also, a lot of stuff isn't showing up here, ground-based measurements of leaf turnover. And what we see here, the tower blew down in 2005 during Hurricane Wilma, so it blew the whole thing down. They put it back up, and we can see a very rapid recovery of a forest that had been completely defoliated. And this forest is incredibly productive, net assimilation up to 1,000 grams per meter square per year, very, very high, equivalent to some of the tropical forest settings that we have known for years are so productive. So this forest is really being enhanced by that steady and sometimes pulsed input of phosphorus that's happening from the marine end of the system. Our groundwater data was showing quite a bit of movement of that saltwater front into the ecosystem. So Renee's data is showing gradual movement of the ecotone between the fresh and marine. And Greg Cook's dissertation work really focused on the effects of that in the lake area where the arrow is pointing in the bottom of Taylor's Slough. And he showed this huge supply of phosphorus coming. This is one of these polar plots showing the values of phosphorus during the different seasons of the year, and the wet season values are quite low and the dry season quite high, and that phosphorus is being mobilized into the water column during the dry season, and he showed a big impact on water column productivity as a result of that pulse. And our consumer research also really highlighting the importance of connectivity along that freshwater to marine ecotone. And Mike Heithause's lab has been studying the movements of alligators and sharks, showing a huge degree of intraspecific variation. So individuals have really different movement patterns, some of them going all the way from the marsh down to the marine waters to feed and then back. So they're important translocators of nutrients. And that individual variation could be really important in allowing these organisms to respond to the high level of variance naturally in this ecosystem, and perhaps the future changes that we expect to see in the level of connectedness in this ecosystem. So moving into FCE III, our proposal for the next 6 years of support went in 2012, and we found out some great news last summer. And so we're moving forward with this new kind of programmatic focus that is a little bit more prospectively looking at what sea level rise is going to be doing to this landscape, over the long haul. And so we've all seen these images. This is Hal Wamless's depiction, just with a bathtub model of what the Everglades may look like, not too far into the future. Over a time frame that we may expect to be continuing LTER research. So we can make some projections. The neat thing about LTER programs is that you can make projections over really long periods of time and actually test them, right? And so we have some ideas about how sea level rise is going to influence this ecosystem that we're hoping, well, I don't know, we don't hope play out in the way that we're hypothesizing because of the impact that we all know sea level rise could have down here. But hypotheses that we can test out over those years. So again, here we have our the two figures that I showed you before with what we expected, or what we found the ecotone to look like today, what we expect it to look like if freshwater is restored, but we need to recognize the important pressures coming from the marine end that we're already seeing impacts of that may not, where freshwater restoration may not be sufficient to push that seawater back, and we may be expecting movement to the interior of this productivity peak. So our central question is really about how climate change and resource management decisions are influencing freshwater availability, ecosystem dynamics, and the value and utilization of ecosystem services by people living in South Florida. So playing up that marine end a little bit more than we had in the past. Our conceptual model is kind of complex, but I'll walk you through the major parts of it, zooming in first on that top layer of the external drivers. So we recognize that our external drivers are both global and regional. They're both climate driven and economically driven. So we have those interactions in this upper box that is affecting the degree of freshwater moving into our natural landscapes and the degree to which marine pressures may be felt. And this all connects up with this local processes box that includes those gradients from the freshwater to the marine in our Sharp and Taylor sloughs in the Everglades, and what's going on in the South Florida urban gradient as far as decisions that are both informed by and very much affecting the shape of this gradient. I have these little circles here in the marsh, ecotone, and marine areas, and that's meant to represent the carbon cycle from the perspective of those 4 core areas that we study: the organic matter and the trophic dynamics. At the bottom of this figure, we have a little image of the same diagram that we want legacies or historical changes in connectivity between the upper and lower boxes and future changes. So we have a lot of modeling work going on, perspective analyses of how our system may change given changes in those drivers. These studies are all being conducted within, again, working group thematic areas, and these are the 4 themes that are kind of referenced here by the numbers in the boxes. So we're studying movement and balance of fresh and marine supplies of water, carbon dynamics, legacy effects. So how have the changes in our landscape over long periods of time, how do they affect the ways in which our ecosystem today reacts to change? And then the scenarios work looking forward. And our core areas and cross-cutting themes are pretty much the same as they were, although we've added this carbon cycling one and put a little bit more emphasis on landscape dynamics within the built area of mining. So preliminary findings, we're just now putting together our annual report for the first year of funding. So I thought I'd just go over some findings in the core areas, beginning with the biogeochemical cycling, looking at how the balance of fresh and marine water supplies influence nutrient and carbon cycling. A lot of this work, well, we're using our long-term data series, of course, that are really rich in biogeochemical data, but also experimental work down in the Florida Bay and Regency Science Center mesocosms, pictured here on the right. We had an experiment down there run by Steve Davis. Oh, I put the working group leadership in the corner of every slide. So if you're considering joining FCE or if you're new to the program, these are the folks to communicate to in your different subject areas. So Steve Davis led a little, and Tiffany Trexler led a little project down in the mesocosms in 2011, where they mimic, they mimic sea level rise by changing inundation and salinity, and looked at response in peat soils from the SRS6 ecotone. And they found that increased salinities, especially when hydro periods were reduced, really motivated CO2 to move out of the peat soils. So this is sort of addressing the degree to which respiratory processes may lead to peat collapse or peat loss, because we're very concerned about the balance between peat gain, right, from productivity of mangroves to what's going on in the soils and the losses that may be experienced as a result of changing inundation and increased salinity. This summer, Jen Kamenowski led a project down in the same mesocosms that incorporated a plant soil system. So we have these little mangrove plants growing in the same peats. And Shelby Survey, a new graduate student, and Julio Perchon and RU we're looking at the effects of phosphorus. So again, we expect phosphorus to be moving in with that seawater as it moves to the interior. So they mimic that as well as leaf loss that would be associated with big storms coming through, and looked at effects on metabolism, growth rates, microbial activity in the plant soil system. The primary producer group, led by Tiffany Trexler and Victor Rivera-Monroy, is looking at these same drivers and how they play out in terms of primary producer community structure and productivity. And they're also conducting work in the mesocosms, looking at plant responses. Tiffany has really long-term data sets from Taylor Slough, and she's been able to show really strong effects of changing freshwater flow into the ecotone of Taylor Slough on Elaeocaris and Cladium relative abundances, productivity, turnover. And the Elaeocaris likes deeper water, Cladium likes more shallow and shorter hydro periods. And so we're seeing a lot of change there as a little bit more water is coming through. But we also have enhanced salinities happening during the dry season in the ecotone, and Cladium is being, abundances there are being really reduced by that. They can't even stand a couple of days of 1 or 2 parts per thousand. So very small changes are affecting these communities. The organic matter dynamics group is really trying to better characterize or dissolved in particulate organic matter moving through the system. We're realizing that this is a really important missing component in our carbon budget, understanding the fluxes and movements of this organic matter. And Rudolph's group in particular has done an incredible job characterizing that organic matter in order to try to help figure out where is it coming from, where is it moving to. And so this is just showing the difference in signatures in the POM and DOM and the degree to which they're broken down in the water column. And our DOM tends to be really highly humic and refractory and not a very good fuel for bacterial activity. So that's really important to know. He's also doing some really neat work on black carbon. We have a lot of black carbon in the Everglades, and it's this wonderful terrestrial signal of fires upstream. The trophic dynamics group continues to work on alligators and sharks. A lot of other fish species are being tagged and monitored, and their movement behaviors studied. This is just showing the difference in the movements of 2 alligators. One is liking to remain more toward the river mouth, and the other moving all the way back into the Everglades to feed and back out to the marine. So this individual variation is a big focus of what they're doing, trying to characterize that variance. This water thematic group, led by Renee Price and Rinku Roy Chowdhury, is trying to connect land and water use and decisions about that in this urban gradient to water movement in the interior of the Everglades. And Shimon Witowinski is a big collaborator in this group. He's been using remote imagery to look at the degree to which fresh and marine water move around in the system and how the length of time that they're sitting on the landscape. And he's really been able to pinpoint the movement of tidal water in and out and where it goes, which is really, really hard to do and really important for the models that we're developing. The carbon group has made some big advances. So the goal of this group is to put together a budget that is dynamic in space and time, filling in some of those holes, particularly regarding organic matter lateral transport. Jordan Barr has made these incredible graphical models of what we need to know about the system in order to do a budget. And Tiffany Truxler has put together all of the numbers that we have so far to try to understand what's missing. And so there's a really cool paper in a new issue of Oceanography that is all about our carbon budget work, where we are and where we need to head. The legacies group is also focusing, like the water group, on connecting water and land use to dynamics within the Everglades system. And Henry Briseno has got these wonderful long-term data sets on our biogeochemical dynamics. Jeff Onstead is leading work on land and water use changes, and the two of them together have big plans for connecting these in a spatially really explicit way. So they've got GIS data sets that they're bringing in on how people are reacting to zoning changes. So, huge scale land use decisions, and linking those to what's going on with water movement, both historically and according to current decision making. I should mention that we have a whole new group of water policy folks who are really trying to understand, they're political ecologists looking at the root of decision making and when those decisions are and are not informed by what's going on within the Everglades landscape. And our last group is the scenarios group, led by Mark Raines and Carl Fitz. We're using a scenario framework that was developed by the LTR network, so a lot of sites are doing this in concert so that we can have some projections of what our ecosystems may look like under various starting scenarios, and be able to compare those across place. And a lot of the modeling efforts within FCE are really mathematical, I should say. We're doing a lot of quantitative modeling, both hydrodynamic and coupling hydrodynamic and process-based models, a lot of which have been around for a long time. So capitalizing on big modeling efforts that have been going on in South Florida for a while. And there's a big effort to bring these models together to help us understand one key thing, which is how that ecotone location is going to shift over time with various driving conditions. So let's see. That's a lot of the background of what is going on within the FCE domain. Again, we do really care about linking our work to other coastal sites, to other similar wetlands globally. And so we wanted to mention just a few projects, a few examples. Right now, I guess we have 16 ongoing studies involving other LTR sites within the U.S. and more broadly globally. Rudolph Jaffe's group has been doing a lot of examinations of the quality of dissolved organic carbon in FCE relative to other places. And what he's been able to show, this is FCE and the purple triangles, and then all these other LTR sites here and the other symbols. And we can see this huge variance in VOC quantity and quality within FCE that almost spans that range of what we see in other sites. So we have an incredible gradient from the freshwater to the marine that deserves the big focus that we give it, because it can give us such a better understanding of how carbon moves around the ecosystem. A big open area of research right now. Jim Forkran's group has also been doing a lot of cross-site research and synthesis, looking at carbon stocks in seagrass beds, and has a recent paper showing that the carbon storage in the soils underneath seagrass beds is almost as great as what we find in tropical forests. So just like our mangrove forests are incredibly productive, we're showing that our seagrass communities are as well, bearing a lot of blue carbon. A lot of our cross-site studies are trying to understand the Everglades in the context of other karst wetlands. So a lot of what I've told you about this, what we call upside-down estuary idea that the nutrients are coming from the marine end, not the upstream, is a result of being in this karst landscape where nutrients are sequestered by all that carbonate. So in order to really understand that and how it matters to understanding the future of coastal wetlands more generally, we've really capitalized on the fact that we have other landscapes like that in the Caribbean. So we have a lot of research going on in the Yucatan Peninsula, from hydrological to food web structure work to producer dynamics. And I just wanted to show you, oh, geez, this is, I don't know why my slides are not showing up. Some interface between Excel and this. Weird. Okay, I'm just going to skip over that. So let's see. Our cross-site collaborations extend into the social sciences. There have been some workshops that have been funded by the LTR Network Office to really drive comparisons of land and water use across sites and how those matter to LTR landscapes. And finally, let me just mention a few things going on in our education programming and our outreach. I don't know what time is getting to be.
It's 1:47.
Okay. I will end with a few things about our graduate organization, our education programming, and a little bit about outreach. We have a huge graduate student organization. A lot of you guys are already members. They're really, really active, one of the most active groups in the network, and our grad students have been running a lot of the network level programming, and we're really, really proud of them for it. So they're doing a lot of network science planning, meeting leadership. They have their blog, videos, reading groups, different outreach activities, socials, a great group to be involved with if you're new to the program. We have our LTER Schoolyard that, again, is reaching out into the K-12 community. Our high school kids are doing fantastically well at science fair. Lucia has had a number of high school students in her group who have just done incredible work and moved on into undergraduate programs with scholarships and wonderful levels of science support. And our outreach programming, we have been trying to move our discoveries into decision making by really capitalizing on the fact that we have a lot of decision makers within our collaboratorship. And so this is just one example of the kinds of ways we get our science out there. We report our long-term findings to the system-wide indicators for Everglades restoration reporting, to the system status report that's put out every year or every couple of years by the Saltwater Water Management District. And this is just an example from the paraphyton end of things that I've been working on, showing where we have problems in this kind of stoplight system of green is good and red is bad. And this goes right up to Congress, and there's actually, get feedback, and that's pretty amazing. I don't know too many cases where science moves as quickly into decision making as in South Florida. That gives us a lot of opportunity. And so our communications broadly are really important for that reason and to connect with other scientists. So we do that through our newsletter, News from the Sloughs. We have artists involved in the program, LPE Art. They're doing all kinds of things to connect the public to science through different artistic means. We have our graduate student blog, a predator tracker where you can follow the predators around in the ecotone online. Various virtual tours and videos that give you a feel for what the landscape looks like if you're new to it, and our new little children's book that's really incredible, and I have a copy of it here, and it's in the LTR office for sale. One Night in the Everglades, reaching the little kids. So I just wanted to briefly mention our information management system, which is really well run by Linda Powell, and you probably get a lot of emails from her saying, please send us your data. Do that. It's really important, not only because we're obligated by NSF to do it, but it is how our information moves itself out to the public. Grad students can wait till stuff is published. Everybody can wait till stuff is published, but it needs to eventually get there so that it can be more broadly used. And if you have questions, I'll have a few minutes now, but our LTR office is right downstairs in ECS 262. Mike Ruby is our program manager and incredibly knowledgeable about every aspect of the program. And our website has a lot of information there too about how to join, opportunities for students, research, discoveries, and findings all in a nutshell. So I'll take questions now, and thanks so much for your attention, and I hope the graphics move through okay. Tell if there's questions.
Yeah, I'll be able to see that there's any chat stuff here.
Maybe you put a light behind it. Any questions?
Evelyn, this is Rudolph. I have a comment. You probably misspoke, but the children's book is not for sale.
Oh, okay.
These are free copies. It doesn't mean that everybody can just show up and get one. They're supposed to be distributed throughout the school system in South Florida, and we've started doing that. It's going a little bit slow because of the way we want it done in an organized way. And if there's anybody who needs a copy, If you could justify it, we can give you a free copy, or you can buy them on Amazon. But the ones that we have are already paid for, so we cannot sell them.
Thanks for
So
that.
I just wanted to clarify that.
Excellent.
Okay.
And I just had a few other things, our brochure, so if anybody wants just, summarized information. And there's more of those in the LTR office. And our special issue of oceanography that focuses on coastal LTR ecology just came out. There's a whole ton of these down free in the office. And we have 3 papers in here, and there's a whole bunch more that are really relevant to what we're doing. Questions? Anything about how to get involved or what else we have going on?
I think I'd just like to make a comment that I think you did a really great job summarizing a lot of facets of the program that's super dynamic. I've been in the LTR as a graduate student since 2003 at Coweta LTR. And that's a great site, but this is a fantastic site in terms of grad student involvement. And as Evelyn said, most of the graduate students are the heavy lifters in FCE. And as she also said, the research findings making a direct impact on management, local management, and federal management is huge. So it's an exciting place to be.
We're excited you're here. Any questions from the WSP perspective or ideas about integration?
Well, it's fascinating. I mean, you have the huge advantage of having been at it for a long time, basically.
Yeah.
And the production of stuff is just remarkable. And also the graduate student, the motivation and everything, just the number of them is so great. And it's a bit daunting, I guess it looks to me at the moment. But so, yeah, I mean, obviously we're covering a lot of the same issues. We're curious about the same issues, and we're trying to address some of the same questions. So there's a lot of room for collaboration.
Mm-hmm.
So we'll continue to try to talk, and this is the first step in trying to do that.
Yeah, great. Cool. All right, thanks everyone for coming. Thanks to everybody who helped with the various means of communicating today. So Christy with the Ustream and lots of help from Alejandro with the Adobe Connect, a little bit of everything.
All right. Well, thank you.
Yeah, thank you.